Okay, I'm gonna start everything off by just taking us into the very basics of what it is we're talking about, the rat lungworm, because we use that term to refer to both the disease itself, rat lungworm disease, but also the parasite that causes that disease. So the parasite is actually called Angiostrongylus cantonensis, that's its Latin name, and the disease is angiostrongylliasis, right? So there's a relationship, but they are two different things. And so we kind of want to break it down and understand a little better so that we can then interfere with this life cycle and hopefully protect ourselves. So the life cycle, as we know, it's called rat lungworm, right? This parasite lives in rats, but it has to spend a portion of its time in a slug or a snail. It has to spend a portion of time in an intermediate host and then return to its original host, or its definitive host, which is generally a rat, in order to fully complete the life cycle. And when we talk about life cycle, that's from birth, or if you're an egg, an egg to hatching, all the way to reproduction. So the life cycle goes to when you reproduce. And the way that this particular parasite lives, it needs to be in two hosts in order to get through all of the stages of life, from being hatched out of an egg to being able to reproduce and lay more eggs. So the way that this works in nature is that the rats find snails and slugs to be tasty snacks. So they eat a snail and a slug, and then the snails and slugs will then eat rat droppings. And the parasite is moving through all of that. It starts with that. It starts with a love story. Every birth starts with a love story. And you're seeing a love story happening right in front of your eyes. That white and red striped candy cane worm, that is actually the female worm. The white is all of her eggs, and the red is the blood that's being taken from the host. And then to sort of the left here, as the screen moves, you see just sort of that dark red that's over here to the side. That's the male. That's the male worm, and these guys are mating in the heart because that's where love begins. So this is what adult nematode rat lungworms look like. And thanks to the Jarvie Lab for providing this very cool video. Where you get into problems is that once those eggs are laid, their mating happens in the heart, the eggs are laid in the lungs. The rat will then have this feeling of something in its lungs. And if you have an irritant in your lungs, you cough it up, right? You got to get that out of your lungs. When the rat does that, the eggs will come into the rat's mouth. The rat will swallow the eggs. And now the eggs are in the digestive system. Once they pass through the digestive system and they get into the feces, that is where they're going to come into contact with a slug or snail that's going to come along and pick that up by snacking on that feces. And then it's going to develop to the third stage inside the slug or snail. This third stage larva goes dormant inside of the slug or snail, and it curls up. So if you see that lower left cube here, how it's curled up, that's the dormancy stage. And you can see a bunch of them in the upper left, right? These guys are just waiting until such time as they are picked up by a rat. And then the hydrochloric acid in the rat's stomach, which is the same type of acid that we have because we're also mammals, that's going to be a sign to that worm to wake up and start wiggling around to get through the lining and get into the bloodstream to continue its life cycle. So that third stage, when it gets into a slug or snail, that's where it stays. It doesn't move on in its life cycle until it gets back into the rat. That is also the most dangerous time for us. Anything that can hold those larvae during that time and allow them to get to the third stage that is not a slug or a snail, and there are a few things that do allow this, are called paratenic hosts. So these paratenic hosts, we know that they're around the world. There are some there are different insects, right, like centipedes. There are flatworms that will hold them. We know that coqui frogs now hold them in their tissues. Prawns, so the Tahitian prawns that we catch, in the freshwater streams here. These things can all be paratenic hosts that could stand in for a slug or snail. There can also be stand-ins for the rat portion of this, right? So as we know that these people can get this, dogs can get this, horses can get this. So some animals seem really susceptible. Some animals seem not as susceptible to rat lungworm. Like there are birds that are affected, and then there are ducks and chickens that don't seem to be affected at all. So there's sensitivities to different animals depending on their digestive system. But dead end host, that doesn't mean it's a dead end for the host. It's a dead end for the parasite. So theoretically, once the parasite gets into one of these animals instead of a rat, then it no longer can continue its life cycle, although that is a little bit up for debate. We know dogs are particularly sensitive. Cats do get rat lungworm, but seem to recover a little bit faster than dogs. A lot of dogs and horses have actually lost their lives to rat lungworm. So the background of what we know about rat lungworm starts in the region of China known now as Guangzhou, and back in the 1930s, it was known as Canton. That's where we get that name, Angiostrongylus cantonensis. So in the 1930s, this was the first time that the disease was observed. And then a postmortem indicated the worms were in the brain. So they were able to actually say, oh, wow, so this disease was caused by these worms in the brain. We saw in the 1930s and 40s lots of movement around the world, particularly during World War II, and there were introductions of giant African snails and other slugs and snails, and of course rats on ships, right? So as you have a lot of things moving across the world, you have a lot of invasive species that go along with all of our human stuff. And the first African snail, it was introduced probably multiple times, but the first time African snails were introduced to Hawaii that we have a record of this introduction was that a woman brought them in her suitcase, got off the boat, and put them in her garden because she thought they were pretty. So that was our first introduction, and that's kind of a common story for invasive species. But the first cases of rat lungworm that we know of in Hawaii were caused by rat lungworm were in 1959 and 1960. So we know that the parasite was here as early as the 1950s because that was two men in Oahu who both died from the disease, and a postmortem showed the worms in their brains. So that was something that we know it's been in Hawaii a really long time. And then, of course, we've been seeing this continued spread around the world. And we now have rat lungworm detected in multiple states on the US mainland. Now, Kelley's going to talk a little bit about this later, but the only thing I really want you to take away from this is it's just a timeline from 2007 to 2017. You see over this 10-year period, there's kind of this real jump, right? There's just a jump in cases of rat lungworm. And we really want to think when you see something like that happening, why is that happening? What we think on the, from looking at this through an invasive species lens, if you've had this parasite around since the 1950s, right, but you don't see it spreading a lot, and then all of a sudden you see it in a 10-year period, this massive spread, something has changed in the environment. And what we think the thing is that changed in the environment in that time period was the introduction of this invasive slug, the semi-slug, Parmarian martensi, right? This was first found in Oahu in the mid-90s. And the first time it was collected on the Big Island was 2004, but most people were saying, when that study was done, that they were actually doing the collections, people had been seeing it for at least a couple of years. So it came in the early 2000s. By 2010 was popping up everywhere in Puna. I literally remember this happening at my house in Puna, where I was like, what is that thing? And then in 2018, it was detected in Maui. That was because there were some severe cases that popped up in Maui. And some of you who followed rat lungworm for a while will know the name Kay Howe. So Kay was a researcher at the Jarvie lab. Her son had a very severe case of rat lungworm. And we were working together with Wendy on a teacher program. And when it popped up in Maui, Kay said, I really think semi-slug is in Maui. I think that's what it is. And she put it on her Facebook, and we all shared it on our Facebook. And within less than 24 hours, we had our first confirmed sighting of semi-slug in Maui, that we confirmed it was in Maui. And then in 2019, one of our teachers from North Kohala had taken the students out to do a slug hunt and sent this very picture that you're seeing on your screen into us and said, my kids found a weird one today. What do you guys, what is this? And then we knew that semi-slug was in Kohala. It had never been reported in Kohala before. So we had two really severe cases of rat lungworm pop up in Kohala that same month. So we see this increase in disease that kind of follows with this appearance of this particular species. So why do we think that is? So one of the things is that we think that the semi-slug is attracted to human activity. So most slugs, they're out in the garden, right? One of the things that happens with semi-slug, and again, if you live in Puna like I do, you may have noticed this happen, is that at night, these guys will actually come to you if you're outside. You'll find them. I actually had a barbecue once at my house, and one of my guests he got up and ran to the bathroom, and he came back and he said a semi-slug was crawling up his leg. So like very, very creepy. But I'll often go outside at night and I'll put my hand on, to take my shoes off or put my shoes on. I'll put my hand on the railing, and my fingers will touch a slug that's like underneath the railing. Like they come into human areas in a way that is unusual for slugs. And in 2017, there was a family that contracted rat lungworm after these slugs drowned in some kava that was being prepared in the backyard. So, that's something that obviously people didn't expect to happen, but these guys were attracted. Why were they attracted to kava? It doesn't seem like something slugs would be into, but these guys are attracted to human activity. So that's one risk factor. The other is that relative to other slugs, they're pretty fast. So, this is obviously sped up, but this is a Cuban slug on the top and then a semi-slug on the bottom. And you can just see comparatively that the semi-slug is much faster than the Cuban slug. They're just moving through their environment faster. And again, if you're outside in an area that has semi-slugs, you can see this happen, where you might have plates out, and as evening falls, all of a sudden you're looking at a plate and here's a semi-slug crawling up the table. They're good climbers, and they can kind of do this like Tom Cruise in Mission Impossible thing where they can drop down. They can make a mucus thread and lower themselves into a new area, so they can kind of overcome barriers that way. And they're very gregarious, so they cluster together. This is in my own backyard, and you can see 1, 2, 3, 4 slugs, and I think there was actually 6 altogether that for some reason were all clustered together. And it's theorized that they use this clustering as a way to get through dry periods, that they can cluster together and make a mucus ball to help keep themselves damp so they don't dry out. And then one of the final high risk factors is that just for some reason, this particular slug is really susceptible to the actual rat lungworm nematode. So in semi-slugs, this was a study that was done, I think the early 2000s, like mid to late aughts, of what the infection rates were in the actually, it was a little later than that, like around 2014, what the infections rates were in the different slugs and snails that were collected on the Big Island. And what they found was that these semi-slugs were over 70% infected. When you just zero in on the Puna and greater Hilo area, it's over 90% infected. So most of the slugs that they collected, if it was a semi-slug, it was almost guaranteed to actually be infected. And then when you get to the other species, it's about a third of them, and then even smaller. Any slug or snail can carry it, but its highest prevalence is in the semi-slug. So just being aware. Unfortunately, even some of our endangered native snails have been found to have rat lungworm in them. But any slug or snail is capable of carrying the parasite. It's just that these ones are almost always infected with rat lungworm, and they just come into contact with humans in a much more often way than the rest of these guys. So what can you do to protect yourself? And Wendy's going to talk more about this, actually like what you can do in the garden, but you can think about around your house, just practicing safe behaviors. And one of the things I mentioned dogs before, and I just want to emphasize, to really think about our animals, because they are very susceptible to rat lungworm. Dogs in particular, because, when you're putting a bowl of food out, there's a lot of protein for our dogs and our cats. When you put those bowls of food outside, if it's evening time, if it's after dusk, when these semi-slugs are moving, again, they're very fast. They're looking for a protein source. They can get right into that bowl, and dogs just eat. They might not even notice that they just ate a slug. So using heartworm medicine can be very protective. It doesn't mean that the dog is not going to get rat lungworm if it eats a slug, but it can really help with the recovery. And so keeping your heartworm meds up to date, and also just not putting food outside at night, or pulling those food bowls back at night so that your animals are not susceptible to accidentally eating a slug or snail. And then a lot of people, especially in Puna, are always very concerned about catchment. The most important thing you can do with catchment, because and Kelley could talk about this, there's a lot of other waterborne diseases that you can get from catchment. So keeping your catchment clean and keeping your whole filtration system well maintained is number one. People are always looking for like, what's the perfect catchment system? It's like, you know, start with step one, which is whatever catchment system you have, make sure it's clean and make sure your filtering is up to date. Your filter is within its lifetime, change those filters as they're supposed to be. And that's really important. When slugs drown, particularly these semi-slugs, when they drown, the rat lungworm parasite, that nematode, is released into the water. And that can continue, sorry about that, that can continue to be a problem for at least a week, maybe a lot more days than that. I think Kay Howe had found something like 21 days that there were still some that were alive in the water. So you want to be really careful with your drinking water. Don't just drink out of an unfiltered catchment. Really, really important to think about that. The Jarvie lab did test some filtration systems and found that some were a little better than others. This 5 micron carbon block did really well. Again, this is a small test, so we can't talk about every single type of filtration system, but keeping that filtration system maintained is really going to help you. So I think we have some in the chat. There's going to be some links that you can click on to go to the Jarvie lab and learn more about the work that they've done on rat lungworm. One of the other pieces that, they've investigated, because there's been a lot of questions about this, is slime. Is there parasites in the slime? Yes, there are parasites in the slime. Only a few, not as much as you're going to get if you're going to ingest. And I take this directly from this slide. This is Dr. Argon Steele, who's a researcher at the Jarvie lab. And I just thought, well, I'm going to take a picture of the slide and I'm just going to share it to show that this is like what scientists are sharing with other scientists, right? There's only a few nematodes observed, so the role is considered minor, but there is larva, L3 larva, in the slime, so it can potentially be ingested. We don't know how much larva you need to actually ingest to get a severe infection, but there are a lot of folks who swear there's no other way that they could have contracted rat lungworm. So it's still an unknown, but it's best to be careful. It's important to know that once slime dries out, they die. They cannot, the larvae cannot be dried out. So once something's dry, if you see slime on the pavement and the sun comes out and it dries up, don't worry. There's not a bunch of nematodes crawling around on that pavement. Once they're dry, they're dead. But you do want to be careful if you're washing your vegetables in the sink and you feel slime, just toss that. Just don't mess with it. Okay, people want to know, how do I clean my vegetables? This has been tested. None of these things work. So I see this a lot on Facebook, apple cider vinegar. It does not work to kill the nematodes. Remember, they get woken up by hydrochloric acid, which is a very strong acid. And apple cider vinegar is a very weak acid. So all this does is kind of rouse them from their nap a little. So that's not going to work. You don't want that. Hydrogen peroxide doesn't work. Seawater, alcohol, neem, all of these things. There are no commercially available, like over-the-counter washes that will work. There are some like vegetable washes that are industrial level that will work, but they're not available for folks to just buy. They're for big farms. Bleach does work, but we don't generally bleach our vegetables, so it's not the best. But yeah, so we want to just practice a lot of protective stuff. And I'm going to pass it off now to Wendy, because I talked about a lot of indoor things, and Wendy's going to get into how to protect yourself in the yard. Are there any questions I need to answer now, Molly, before we move on, or anything that's specific to the parasite? I think I see a lot of
Comments.
okay.
Let's cover our drinks when we're outside. That's always a good one. I'm going to go change my catchment filter, maybe when Wendy's speaking, or set a reminder to do it later. How is it possible that someone could have rat lungworm in 2013, be dewormed, it goes away, then get sick again in 2026? Is it dormancy? Because what are the odds of two people in different households get sick at the same time exactly 7 years later?
Well, I'm not going to answer the health questions.
Okay.
I can say that you can get reinfected with rat lungworm. It's not the type of thing where you're going to build up an immunity like measles or something and then have that immunity. Like it's if you get rat lungworm and then you eat a slug 7 years later, you can get it
Yep.
again. But I'll let Kelley answer more of the health questions. I will answer the one that says, will UV filters kill the
Yes.
larvae? They damage it. So UV is really good for single cell organisms. And this is a very complex multicellular organism. So UV can kill some of the nematodes and it can damage the nematodes and slow them down. Just like UV damages our DNA, the UV can damage their DNA, but it is not 100% kill. So we usually tell people you'll want, different stages. And I think UV is great. Again, other diseases are very common in catchment. So you should have a UV if you can afford it. But yeah, you want to have different stages. You still want to have the other filtration system. Okay.
Great.
I'll pass
Thank you
that
for
off then to
telling
Wendy.
us ways to reduce our risk for rat lungworm.
Yes.
Awesome. Thank you, Franny. Thank you, Molly. Aloha mai kako. I'm Wendy Baker, and I'm coming to you from Pahoa, here at Hawaii Academy of Arts and Science Public Charter School, where I am a garden teacher. And as a garden teacher, it's my mission to create harmonious learning garden environments where students happily nurture healthy habits. Oh, there's the bell. I'm at school. Awesome. Thank you. As in any classroom, safety is fundamental in realizing my mission. So, an excellent way to make sure that we are having a safe, harmonious environment is through practicing integrated pest management. And IPM is a systematic approach that creates balanced ecosystem using organic methods, usually organic methods that optimize the health and resiliency of an ecosystem while mitigating damaging pests. We're going to look at a couple different best practices for mitigating rat lungworm, and I will share my slides. Let's see. There it is. Let's try this. And I'm gonna get rid of this one. Sorry about that. Here we go. Oh. Is it all right if no, you guys want to see it bigger.
Yeah, I can see it. And then just go to if you go to present at the top, there should be I think
Yeah,
it's
I was
that.
looking.
Yeah, click on that one.
There we go.
There.
Better. Okay.
There you go.
Excellent. Thank you. All right. So just an overview of angiostrangulosis and the practicing of safe behaviors. So just today was in the garden right before coming here and telling the students, sorry guys, I'm gonna have to cut out for a little bit. So they're with another teacher. But we were picking spinach. The kids are making a couple different dishes today, some crepes and savory crepes, and they were filling them with spinach. And so we were washing every leaf, and then we lay every leaf out on the stainless steel table on a white towel, and we found a little slug, semi-slug, like the size of your pinky, like the tip of your pinky, very small. And so that's part of it is just teaching kids like, okay, we got to practice safe behaviors, and then we're cooking everything. So ultimately, it's not a problem. But I did take the opportunity to remind everybody about why we take so much time and how we have to be so careful when we're inspecting our produce, right? So that's one safe behavior is just to really remain mindful and to pay attention to what we're doing and to what's going on around us in our environment. One way we can do that is by checking for this guy. Yeah. Checking for slugs and snails. You can find them in many different places, like on leafy greens. They love hard plastic. So we have a compost container that's a hard plastic one. And so we're always checking there for them. And the garden hoses. So all of our garden hoses here at school have a spray nozzle at the end, and they're always wrapped up and put up high so that this isn't going to happen, so that the slugs will like to crawl into places like Franny says. They like to be around people. So we try to manage our habitat. So that means that we're clearing vegetation and doing raised garden beds. So trying to keep them out. They'll still crawl up, just like Franny said, or I guess they'll drop down, Franny. I didn't know that they could create a mucus slime like a spider and drop down. That's crazy to me. Yeah. But really mitigating your environment. So looking around your environment, managing it, and limiting habitat is where you want to go. So this is where clearing old vegetation and getting food up off of the ground. Yeah. And then this is a solution to deter slugs and snails. Um, I haven't had luck with that, so, but for treating the raised beds, I pretty much have gone away from this method of raising beds and just use Sluggo, which we'll get to in a minute. Uh, we always have a slug jug. It's got 1 part salt to 6 parts water, more or less. Great ratio lesson for kids. So for them to understand ratios, always rational numbers, ratios is always a good thing to work on for kids. And whenever we find them, I'm the one that does it. I just go and I get the slug jug and I'll put the slug into the jug. Yeah. But the kids all know to keep an eye open for them and to jug them when we see them. Yeah. We hunt early in the morning or during wet weather, and you have to make sure that the lid can close tightly. The semis, if they get out of the water, if they reach the side, they can crawl up and nest along the top of the rim. So you do want to be careful about that. At home, I use diatomaceous earth in just a yogurt container. That's good enough for them. And at home, I've noticed that uh, there seems to be little seasons, and I haven't figured out yet if they correlate to any particular time of the month or year. But there are times when there's like a kahea, a call. It's like, hey, come on, guys, everybody come out. Let's do our little our romantic mating orgy thing that we do. And they all come and cluster. And so that's a really optimal time, is just to be observant and walk your perimeters and check. And so I notice that if I'll see one day, then the next day there'll be like 7. And then maybe the next day there'll be 12. And then the next day after I jug them all, there'll be none. So that's just being, again, managing your environment and being aware of what's happening around you. Yeah. Let's see, you always want to avoid touching slugs and snails with your bare hands, obviously using gloves, tongs, and or chopsticks. And of course, in the school garden, those are all labeled slug only. Yeah. And then this is a Sluggo that we use at school. And I have to say that when we started this, uh, Kay Howe did a study, maybe 2017, I think, and somewhere around there. And we had huge numbers of semi-slugs. And since I worked the perimeter of the garden with weed mat, and then Sluggo goes down along the perimeter of the garden regularly. And now we have very few semi-slugs. We do still have some African snails in the garden, but fewer and fewer. And rats is another thing. So with integrated pest management and rats, there's all different ways you can control for rats. A rat zapper. This is Franny introduced me to this idea, so she could talk more about that one. We tried to have a garden cat for a while, but he didn't want to stay in the garden, of course. He just wanted to be up where all the kids were, so he had to get adopted and moved away. But since we've had the cat down there, he was there for about 4 months. We haven't had rats back again, so that's been good. Yep, so just practice safe behaviors and keep yourselves safe from rat lungworm. This is a little bit more about the washing, which is how I started the conversation just because of my experience today. So just making sure that we're really being aware, washing each leaf, drying each leaf, and using all of our senses and focus when we are preparing food, and just making sure that that we're not ingesting or missing any of them. There's a food prep video, so if you guys would like to scan that. I did not try this one to make sure that it's up to date, but for any guys, I bet you can drop that into the chat if needed. And then always the sure method to kill all nematodes is to freeze, cook, or boil, heat your food, and just make sure that you guys are staying safe. And then I think we're probably going to talk, I'm not sure, but I'd be happy to talk a little bit about what happens if when you do contract, if you do realize that you have a snail in your mouth, which has happened to me in 2021. The steps to take right away are to take a dewormer, right? So there's a pinworm medicine that you can buy over the counter, but albendazole is really the one that you want to go for, and just seeing your doctor right away and being sure. I was very lucky that the doctors that I encountered were knowledgeable and asked good questions, and then gave no pushback at all, and prescribed to me the medications that I needed, the steroids and albendazole. 5 years later, yep, 5 years later, I'm still struggling with some of the symptoms, mostly neuropathy. So just again, it's a dangerous disease. I'm very lucky that I didn't have a higher worm count or nematode count, and that I was able to manage. And I had amazing helpers that helped me to heal. So on that path still, but let's just really protect our communities. It's a very nasty disease that nobody wants to get. All right. Mahalo. Thank you.
Thank you, Wendy, for sharing your story. I'm glad you're on the path to healing. But yeah, and thank you for sharing this information so everybody in our community can take steps to reduce the risk of contracting rat lungworm disease. Thank you. Okay, Kelley, we'll pass it on to you now.
Okay, thank you. When I first moved here in 2014, one of the first things I was told when I was teaching at the university was about rat lungworm. And, being a nurse and an infection preventionist, it definitely intrigued me. And there wasn't a whole lot of work going on at the time, but starting in 2020, I became part of the support group. And so I started hearing these stories, and, made me certainly want to start doing a little bit more where I can, use my knowledge as an infection preventionist and a nurse to just raise more awareness, along with all these wonderful folks on the call. So let me share my screen. And Shazi were one of those stories I kept hearing about. So, you were my motivation to try to just figure out how can we do a little bit more to raise awareness. Okay, you can see my screen okay. So I am the infection preventionist, which just really means I try to, prevent all kinds of infections in our community. It doesn't have to be rat lungworm. I primarily help support long-term care facilities or, healthcare settings. But I get to do community awareness and work with great partners. I'm just, I was asking Franny before this life cycle, and do parasites normally go through this 2-stage through the rat? And she's like, oh yeah, 3 stages on some parasites. So just kind of an interesting disease and just how it works. And so knowing how it works helps us figure out how to prevent. So this is the number of cases. I took a look actually from Franny's slide, which ended in 2017, and then I took it from 2018 and just did this look back. And so we have confirmed residents and non-residents, so visitors. And the confirmed is really just that there was symptoms and that you ended up getting a lumbar puncture, or these people ended up getting a lumbar puncture that then put them in this confirmed category. And so, there's been task force work back in 2018 when that, 2017 spike happened, and there were some guidelines that came out from our work group. And then in 2022, got some new guidelines from Dr. Ansdell. So we're actually following those guidelines. And the 2018 has been archived. And then, you can see we just had a little bit of a blip. We don't have a whole lot of cases in this confirmed category. We do have a probable category, and those are the ones that may have symptoms but didn't end up getting a lumbar puncture. I know it can sound scary, and so that sometimes is a barrier. But we, definitely direct folks to, if you want to get confirmed and know what it truly is, because anyway, we re-engaged the task force in 2024, and then this new sensitive test came out in 25, and actually Hawaii is the only one that has that new sensitive test, and CDC doesn't even test anymore because they know that we have the better test. So we brought that on board, and then that helped launch this provider toolkit that we have been passing out to all of our providers. There is, some misunderstanding about rat lungworm. We've heard some folks think it's, because there's no cases on Maui or Hawaii for a while that it went away. And just, because we don't have a whole lot of cases, it's not something that providers have on their top of mind all the time. So this was my way, this provider toolkit, to bring it to top of mind. And then also just making sure our community knows about it. What we've found is our community knows about it. They just may need to tweak a little bit about their strategies to reducing slugs and snails. And this is just a map, just to highlight that the east side of Hawaii Island has the most cases. And so really targeting our outreach to that side. But I do have my counterparts on the other islands, they're also doing outreach. And so currently what's going on, it's a multi-organizational collaboration to work on rat lungworm prevention. So we did a major update to our website last year. So I would really encourage folks to go there. There's a couple things that I want to add, but just, I think it's pretty well done, pretty thorough compared to what it was. And then we have that more sensitive PCR test, doing that community and outreach to providers. And then our work group meeting, I'm the lead for the outreach. We have also a research group and then a clinical working group. Department of Education, there's that integrated pest management online course. Big Island Invasive Species also does community outreach. And then you guys, I don't know how active the snail tracking app is anymore, but I know that was used in the very beginning. It's very interesting about those semi-slugs too. I have seen them come down the side of my house, around my window. I had like 10 or 20 of them from all different sizes. And they're actually really kind of hard to get with my tongs. So that was the first time. I should have taken a picture, but anyway. And is that the reason why, Franny, that east side of Hawaii Island has potentially more cases, is because we see more semi-slug on this side of the island? I can't remember if I
Yeah, that's what we think. I mean, we think
Yeah.
it's really, you just see when the semi-slug turns up someplace that's where you get these increased cases, and that's why I go over like why we think that. But yeah, I think it's also just it's damper over here, and they can live longer and prosper more because they need that wet.
Yeah, anyway, I just wanted to make mention because, yeah, they just come from everywhere. And then we have folks doing research activities. It's ongoing, our University of Hawaii partners, and then Bishop Museum, which I didn't know that they did research, but they do. So our community engagement grant, I'll have some pictures about our slug jug starter kits. We were able to procure a 10,000 $10,000 community education grant. So we're having some fun with that, really trying to, again, get out there and talk about strategies to reduce slugs and snails. We go to all kinds of community meetings, summits or fairs. We went to the the block party, Activate Puna block party. It probably wasn't the best place to be passing out slug jugs, but we still did it. And we passed out over 80 provider toolkits. We've been to Pahoa, Hilo, and now we're going to the other side. We have dropped off some to the other side. I was going to say part of the community when we do outreach are those two Big Island invasive species resources here, this making a slug jug, and then we turned these into magnets. So we passed out, I don't know, 5,000 magnets. And then this is Emily. She's our investigator on this side of the island, and this is us preparing these slug jug starter kits. So we were able to get swag bags and put all kinds of resources in them. We started it and put a 1/2 cup salt. So it's a starter kit. And so we do have that recipe on the top. And we have gotten so much great responses from this. One thing that I think was an aha for us is how people are getting rid of slugs and snails. You know, that's still the old way of thinking of, I'm going to go pour salt on the slug. But, you know, that doesn't kill the parasite. So we're reminding folks, you want to kill the parasite. So physically removing it and putting it in a slug jug. The other thing is that you don't want to leave rat food. So if you're just pouring the salt on the slug, now you're creating rat food, or, you know, because it's really a combo of rat management and slug management. So that's been kind of an aha for folks, is you physically need to remove the slug and snail. If you have concerns about the slug and maybe, you know, that you don't want to kill them in this saltwater solution, the other way to be more gentle to the slug, which doesn't sound gentle, but you can boil them or freeze them. So those are other ways that you can, you know, collect your slug and get rid of it. But the idea is that you're physically removing it. And the slug jug, you got to have them in the saltwater solution for 2 days to kill the parasite. And then we tell people to go dump it in a puka or onto some weeds. Some of the resources we pass out, so some of my most favorite, not only the Big Island invasive species ones, but the sea tar control slugs and snails. I think maybe we're going to put that in chat. So we're just taking that one section of the slug jug, but we also try to highlight these other ways that you can, you know, just minimize that load on your property, or just reducing your risk. And then this one from the School of Pharmacy, just reminding, yeah, really clean your vegetables. And, you know, don't be afraid to buy local. You know, I get a lot of questions about, should I just buy mainland? No, you want to buy local, but it's just going to take a lot of time, you know, to really inspect, wash, you know, that check, clean, cook on that magnet. You're going to check, clean, and maybe repeat it a couple times, but it takes a while if you're going to eat a salad here. I'm living in Hawaii. One of our new flyers that I need to still get onto the website is about pets. And so, yes, people tend to get a little, their antenna goes up when we say your dog could get rat lungworm. So this is a new resource, and I could put it in chat, or you can go to our website in about a week. I just need to have it loaded on there. But anyway, just, you know, information about pets. And so those best practices about removing food and water at night. I tend to get lazy. So, you know, just admitting that. So these are always good reminders. And I put in chat homestead management. Somebody had mentioned that in another talk. And so it's just constant living in Hawaii, and it's never going away. So we developed, just to help increase awareness for providers, part of the provider toolkit included a couple new resources. And so this one is just really reminding folks it's here in Hawaii, it's everywhere, it's not going away. And then essentially we're all at risk. If we live in Hawaii, we're at risk for rat lungworm prevention. We wanted to bring forward signs and symptoms for those providers. So just reminding them that incubation period. So how long it can incubate before you showed symptoms could be a few days to greater than 6 weeks. So it's really kind of a wide span. And then that medium time from exposure to symptoms is 1 to 3 weeks. So, you could go to your provider and say, I have a really bad headache, which is often the complaint or the primary complaint, but that could be a lot of different things. And so we put together this table of adults and children, because one year we did have a couple kiddos that ended up getting it. So again, homestead management, just, if your kids are going to be out in the yard, you want to make sure they're not putting anything in their mouth. But we put down these earlier symptoms to symptom progression, and really in adults, you're going to have nausea, vomiting in the beginning, as you ingested that slug. And about half of our cases don't know how they how they got it. So we do go off the assumption that it must have been ingested somewhere, somehow. So once the parasite's going to start moving through the body, then you start having these kind of sensory, neurologic kind of symptoms to where, that itching and just really sensitive to the skin. And then, on to like, I really can't move my arms. I'm now, feeling paralyzed. So it does have a symptom progression. And that's often when people are going to the hospital now and, getting that lumbar puncture. So, we just try to encourage folks, if you're just feeling weird or off, or you think you've, been exposed, just maybe print this out and take it to your doctor. You want to make sure your doctors are well informed, because they could be looking at other things, not just rat lungworm. And then in children, it's kind of a wide variety of symptoms too. But what brings them into their doctor is that aversion to touch or being not wanting to be held. And then we're just reminding folks, or the providers, that you got to send them to the hospital to get a lumbar puncture. And then that sensitive test, we no longer require a certain level. It used to be like 10% eosinophils in that lumbar puncture, but the test is so sensitive, no need for that anymore. And then just reminding the treatments, steroids and albendazole. And once the lumbar puncture happens, you can go ahead and start that medication ideally. Other resources that I like, excuse me, CTAR has some great resources. I mentioned the rat management, so it's really that combo, and they just have a lot of great videos, training videos. And then it is, we do want to remain calm about this. It is the one thing that people really are fascinated and really interested and are scared about. So another reason why I want to try to let's talk about it and prevent it. So just good reminders. Clean your hands after you've been out in the yard, cleaning your produce, managing just that homestead management. And then, yes, you can boil or freeze to kill the parasite. I have some more. Let's see. My computer's frozen. Pause. And I can ask a couple questions. Okay. Oh my gosh, my computer just was freaking out. Hold on. Where was I at? A great resource that's now open to the public. It's available on demand is the PBS accidental host. It's 1 hour. This is an old flyer. So we had it one year where they would, only release it a couple times a year. But now it's available on demand, and I'll put the link in chat. Starting to get a little bit more on social media. So just good reminders too, after the storm, the slugs and snails like to come out a little bit more. And so we put together this preventing rat lungworm after the storm when it was so rainy. I think this was, I don't know, a few months ago. So just those important same tips. And then resources, Dr. Ansdell's guidelines, and then all those departments we mentioned with those great resources. And then, if you have any questions, you can contact me or Emily. She's the investigator. And then feel free to scan that QR code to go to our website. So thank you.